Today I would like to talk to you about some of the work that we do in our lab, and this is really the work of two PhD students in my lab, mostly a talented PhD student in my lab. Her name is Victoire Faure. And really what we try to do in the lab is look at the function of long-encoding RNAs in early development or sometime in cancer. But for today's poster, I would like to show you some of the work we've done on early development, or more specifically, pluripotency, which is using embryonic stem cells. So embryonic stem cells are derived from the inner cell mass at E3.5, and they can give rise to all cell types and the three germ layers: ectoderm, mesoderm, and endoderm. We had identified through a screen a long-encoding RNA that we named Tapir, that's specific in ES cells. And here you can see in this figure here of the read coverage of RNA sequencing of ES cells and differentiated ES cells toward EBRCs, mouse and human differentiated ES cells. And you can see from the read coverage that Tapir is actually downregulated upon differentiation of ES cells, suggesting that it's very specific to expression to ES cells. To study it further, what we decided to do was to use a CRISPR-I to knock it down. Using CRISPR-I, when we knock down Tapir and look at day 5, the expression of the pluripotency genes, we can see that the expression of well-known pluripotency genes called OCT4, NANOG, and TFCP2L1 are really downregulated upon knockdown of Tapir. And when we look at RNA sequencing of these cells, you see it here in the black or dark blue cluster. We see that the cells move from a naive state, which is the cells that we can derive, or ES cells that we can derive from an inner cell mass. It resembles that part of development, towards a more primed pluripotency state, which is very slightly differentiated from ES cells, but still not differentiated all the way. So it suggests that Tapir knockdown causes the exit from the naive pluripotency. And looking deeper into this RNA sequencing data, we also find that a lot of the markers of differentiation are upregulated, and suggesting that, again, Tapir knockdown causes differentiation, and Tapir is required for the maintenance of ES cells. What was interesting also, we found that in some of the keywords that came up was alternative splicing, suggesting that Tapir may play a role in alternative splicing, which I'll come back to in a little bit. By looking at the sequence of Tapir, we identified a sign element, sign B2 element, which is known to be found in about 10% of the genome. And more specifically, when we looked at a lot of the genes, pluripotency genes or chromatin remodeling genes, upon knockdown of Tapir, we didn't see a marked decrease. So because remember I spoke about alternative splicing, we decided to look at different genes in the RNA sequencing and look at if their splicing was affected. So some of the genes like EHMT2, upon knockdown of Tapir, you see a 35% increase in alternative splicing of exon 6, suggesting that Tapir is implicated more in the splicing of the gene rather than the gene expression itself. And this, we see it in multiple other pluripotency genes like Stella and MYC. And we think it's through the hybridization of Tapir with sign element sequences that are found in the RNA. Such as here's an example of REX1 RNA. You can see a very strong homology of an inverted sign element in the REX pre-mRNA. So we also wanted to do a knockout of Tapir. However, when we knock out Tapir, it seemed like it was almost impossible to knock it out unless we add a transgene that expresses Tapir. So here we express Tapir wild type, but also Tapir lacking the sign element. If you look at here at the bar graph, we can see that only when we have Tapir expression in the cells and the knockout, that's when we're able to do a knockout, when we have the transgene of Tapir, whereas if you delete the sign, you don't have this effect. You cannot get a knockout, suggesting that the sign element within Tapir is important. Next, here we've been talking mostly about loss of function. The next thing is we want to see a gain of function. For that, we use the reprogramming, which we reprogram differentiated cells towards iPS cells. And when we do that, when we look at the RNA sequencing of differentiated cells, we see that Tapir expression goes up and remains leveled at ES-IPS state, and this is similar in a reprogramming system of human cells. We also did our own experiment using a system where when the cells become IPS, they turn green, and we use neural precursors and introduce the reprogramming factors, OSKM, OCT4, SOX2, KLF, and c-MYC, with and without Tapir, to see what happens. And as soon as we add this, we can see that a lot of GFP, the reprogramming happens quite quickly when we add Tapir, and we see GFP expression much earlier, which means that more IPS are forming much earlier when we overexpress Tapir versus the control. And here's another graph, another way of showing it, that as early as day 14, we have some green cells, where in the control you don't really have many, and this is much higher even at day 18 of reprogramming. Also, when we remove the sign, we can see that the sign element is required for Tapir because we lose that enhancing effect. Next, we did some RNA sequencing on the reprogramming, and we found that there's a deviation of the Tapir, or the early reprogramming path between control and Tapir overexpression. And when we focus on these time points, especially at day 2, we notice that a lot of the genes implicated in prepotency are activated, but more importantly, some of the chromatin remodeling genes are overexpressed. When we do a pull down in ES cells of Tapir, we found that a lot of these chromatin remodeling genes are pulled down. The RNA of them is pulled down with Tapir, suggesting that Tapir, through chromatin remodeling, enhances the ES cells or the prepotency. We're currently looking into a model where this, through the chromatin remodel, Tapir is really affecting pluripotency. And thank you for your attention.